Hello everyone, and welcome to Lecture 7 of BIOC 301. Today, we'll be diving into the fascinating world of iron-sulfur clusters and their crucial roles in various life activities. As you can see, we'll be exploring different types of these clusters, including 2Fe2S, 3Fe4S, and 4Fe4S. I'm Mohammed Shahid Nadeem from the Biochemistry Department at KAU, and I'm excited to guide you through this topic. Iron-sulfur clusters are essential cofactors in numerous biological processes. These clusters, composed of iron and inorganic sulfur atoms, are arranged in distinct geometric patterns. They are particularly vital for electron transfer, playing key roles in respiration, photosynthesis, DNA repair, and the activation of small molecules. Found within iron-sulfur proteins, they are central to redox reactions, enabling energy transfer within cells. We'll explore how these 2Fe2S, 3Fe4S, and 4Fe4S clusters facilitate these processes. Fe-S clusters are found in a variety of proteins, each with unique structures and functions. We'll be looking at different types, including 1Fe0S, 2Fe2S, 3Fe4S, and even 8Fe7S clusters. Each iron atom within these clusters is coordinated to sulfur atoms and cysteine residues from the protein. These sulfur atoms bridge two iron atoms, stabilizing the cluster structure and enabling efficient electron transfer. Iron-sulfur clusters play a vital role in the electron transport system. As you can see from the diagram, they are integral to Fe-S proteins, which are involved in processes like NADH oxidoreductase and succinate dehydrogenase. These proteins then branch into photosystem 1, XPD helicases, rubridoxin, and ferredoxin. Ultimately, iron-sulfur clusters facilitate the transfer of electrons from donor molecules to acceptor molecules, with mitochondria and chloroplasts being prime examples. The electron transport system is crucial for life, transporting electrons from donor to acceptor molecules. There are two main types, one capturing solar energy in photosynthesis, and the other releasing energy as ATP in respiration. Electron-transferring agents, or transferases, include two key iron-based redox systems, high-spin tetrahedral Fe2 or Fe3 found in rubridoxin and ferredoxin, and low-spin octahedral Fe2, Fe3 found in cytochromes. Let's delve into rubridoxin, a low molecular weight, non-heme, iron-containing sulfur protein. It's unique as an Fe-S protein without any inorganic sulfur, coordinated instead with 4 cysteine residues. We denote it as a Fe10S system. As you can see in the diagrams, the iron center has a tetrahedral geometry, coordinated by these 4 cysteine residues. Rubridoxin acts as a one-electron transfer agent, with Fe2+ maintaining this tetrahedral geometry. Iron plays a critical role as an electron carrier, especially in sulfur metabolizing bacteria and archaea. Remember that low-spin complexes absorb light at shorter wavelengths, while high-spin complexes absorb at longer wavelengths. The oxidation state of the central iron atom shifts between Fe2+, reduced, and Fe3+, oxidized, and in both states, the metal maintains a high-spin configuration. The chemical equations here illustrate the reduction and oxidation processes. Now let's discuss ferredoxins, soluble proteins that catalyze the reduction of NADP+ to NADPH. Ferredoxin moves to carry an electron either to a thylakoid or an enzyme that reduces NADP+. Thylakoid membranes have binding sites for ferredoxin, and its main function is to carry an electron from the Fe-S complex to the enzyme ferredoxin, NADP plus reductase, FNR. Type 1. 2Fe2S ferredoxin is very acidic and has a distorted tetrahedral structure, with each iron atom bound to 2 sulfur atoms of cysteines and 2 inorganic sulfur ions. Here we have the structure of type 2, 4Fe4S ferredoxin. Notice the cubane type structure. 4 iron and 4 sulfur atoms are positioned at the vertices of this cubane structure. In this arrangement, each iron ion is bound to 4 different sulfide ions, S. Type 3Fe4S ferredoxin was first identified in nitrogen-fixing bacteria and also features a cubane-type structure. Interestingly, 3Fe4S and 4Fe4S ferredoxins have similar absorption spectra with a broad peak around 410 nanometers. Ferredoxins are associated with both photosynthesis and respiration. A key difference is that ferredoxin has a considerably lower redox potential compared to rubridoxin. Ferredoxin is a chloroplast protein of plant origin, while rubridoxin is mainly found in bacteria and archaea. Ferredoxin NADP plus reductase, or FNR, is the enzyme that transfers electrons from reduced ferredoxin to NADP plus to produce NADPH. As shown in the equation, this is a reversible reaction. Ferredoxin is a strong reductant that functions in 1-electron reductions, while NADP plus accepts 2 electrons in the form of a hydride. An intermediate molecule is required for this electron transfer, and the process is catalyzed by NAR, or nitrate reductase. This diagram illustrates the electron transfer process involving FAD, or flavin adenine dinucleotide, in ferredoxin NADP plus reductase. FAD accepts electrons from ferredoxin, FD, and then transfers them to NADP plus to produce NADPH. This occurs in 2 steps. FAD accepts 1 electron at a time from ferredoxin, becoming FADH, semiquinone intermediate, and then FADH2 subsequently transfers 2 electrons to NADP plus to produce NADPH. Now, let's move on to NADH dehydrogenase, also known as complex 1, a crucial iron-sulfur protein in cellular respiration. Complex I consists of 42 polypeptides, including FMN containing flavoprotein and at least 6 FES clusters. It's L-shaped, with one end embedded in the inner mitochondrial membrane and the other extending into the matrix. Electrons flow from NADH to ubiquinone, Q, via FMN and these FES clusters, driving the pumping of protons, H+, across the inner mitochondrial membrane. This diagram shows complex Y within the electron transport chain. It illustrates the flow of electrons and protons, starting with NADH and ending with ubiquinol, UQH2. We can see the reduction and oxidation of various molecules like NAD+, FMN, and FES centers. This process also involves the extrusion of protons into the intermembrane space, contributing to the proton gradient. Succinate dehydrogenase, also known as complex 2, is another important FES protein in the mitochondrial electron transport chain. It's located on the matrix side of the mitochondrion and participates in the Krebs cycle as the only membrane-bound enzyme. The diagram shows the flow of electrons and the various components involved, including 2Fe2S, 3Fe4S, 4Fe4S, FMN, and FAD. This diagram provides an overview of the electron transport chain in the mitochondria, highlighting complex 1, complex 2, and complex 3, as well as ubiquinone, Q, and cytochrome C, CTC. Electrons flow from NADH and FADH to oxygen, the final electron acceptor. Complex 2 plays a crucial role, and ubiquinone acts as a mobile electron carrier within the chain. Cytochrome C oxidoreductase, or complex 3, is a key component of the mitochondrial electron transport chain. This diagram illustrates the transfer of electrons from ubiquinol, QH2, to cytochrome C via a series of electron carriers within complex 3. The 2Fe2s cluster is particularly important in facilitating this electron transfer. The process also involves the movement of protons from the mitochondrial matrix to the intermembrane space. Thank you for your attention. I hope you found this lecture on iron-sulfur clusters informative.